So in Chapter 8, we're going to look at microbial metabolism. And so a lot of the concepts and a lot of the terms in this chapter should sound familiar from what you learned in biology about metabolism, about cellular respiration, and things like that. But of course, we're focusing on microbial metabolism here, so we will see a few differences than what we saw with like eukaryotes that we talk about in biology. So this chapter begins on page 222 of your textbook. And so to begin with, the first thing it says is that if we're going to look at microbial metabolism, let's remind ourselves of a couple of things about metabolism. First off, what is it? Well, by definition, it's all the chemical and physical workings of a cell. Some textbooks will say that it's the sum of all the chemical reactions of the cell, or it's the rate of all the chemical reactions in the cell. But if you'll recall from biology, these chemical reactions take place in two types. So you have catabolism and you have anabolism. And so what I usually just tell students to remember is if you catabolize something, you're breaking it apart. It's the breaking down of molecules into smaller molecules. Now, be mindful that anytime you break down a molecule, you're typically going to release energy. Now, the benefit of that is that your cells can then use that energy to anabolize or to build a molecule. And so our metabolism or our chemical processes are actually just a grouping of these two types. You break material down just so that you can build something back up. And as you break that substance down, it gives you the energy to actually build. And again, these are terms that you should have learned in biology, but again, just want to take a moment to kind of go over those. Now, we know that every chemical reaction in our cells, or pretty much every chemical reaction in our cells, is going to require enzymes. Remember, enzymes are what actually speed up a lot of these chemical reactions. Remember, what enzymes do is they take that activation energy. If you recall from biology, that's the amount of energy that's needed to start a reaction. And what enzymes do is they lower that. Because if you can lower the amount of energy you have to get to in order to get a reaction started, well, you can get it started a lot faster. Now, be mindful that this little information on enzymes is not on the objectives list. So this is not testable material, but we did just want to remind you a couple things about enzymes. You know, enzymes are protein by structure, and so shape is very important with them. If they lose their shape, or if they're not built to the right shape, they will not serve their function. So shape is very, very important. Remember, we typically name enzymes by changing the substrate ending to ASE. Right? We also note that enzymes are very substrate specific. Okay, so if we kind of skip forward then, our second objective starts to look at energy and some of the reactions that are used to generate this energy. And so first off, just reminding you of what energy is. So energy, by definition, it's the ability to do work. So what that means is if you want your cells to work, if a microbe wants its cell to work, it's going to require energy. Now, energy can be in the forms of lots of different things. So you can have thermal energy, obviously that's heat, or electrical, mechanical energy. The main energy that we're going to focus on is chemical energy, right? Chemical energy, we talk about the main form of that our cells use, or stored energy, is ATP. Now, two reactions that are typically used in order to generate energy are what we call oxidation or reduction reactions. And so if you look at an oxidation reaction, what typically happens is a chemical substance loses electrons. So what we typically call it is an electron donor. So something that's oxidized has given away an electron. And then the reduction would be the opposite, to gain electrons. So if something is being reduced, it is gaining electrons. We call it the electron acceptor. And so typically these two types of reactions occur as a pair. So when oxidation and reduction are occurring together, we call it a redox reaction. So again, you have one substance being the electron donor, you have the other being the electron acceptor. And so the benefit of this is that energy can be released in the process, and it can be captured because, again, it requires energy for cells to work. So we've mentioned this energy thing a little while, and so want to just remind you of what ATP actually is. So if you look at ATP, it stands for adenosine triphosphate. So it's what we actually call the currency for a cell. So if your cell wants to generate energy, this is what it's going to have to use. And so adenosine triphosphate is basically a three-part molecule. Some textbooks will call it a special type of nucleotide, because if you remember from biology, a nucleotide is made of three things: phosphate, a pentose sugar, and a base. Well, if you look at adenosine triphosphate, you'll notice it's made of actually three phosphates. That's where the tri comes from. It's made of the pentose sugar ribose, which is also the same sugar that's made in RNA, and then it's made of a nitrogenous base known as adenine, which again is one of those bases that we talk about with DNA and RNA. And so if you add those three things together, you build ATP. Now, what's interesting about ATP is that it contains energy. It's a form of stored energy. So if you can take ATP and you can break it apart, you can release the energy that's inside of it. And so that's what actually happens in our cells, is that we take ATP, and as we break it down, we're able to generate energy. So there's three basic mechanisms that are used in order to generate energy. The first one's called substrate level phosphorylation. Now it sounds really fancy, but this idea of phosphorylation is just ways that we generate ATP. Phosphorylation means to build ATP. And so if you look at the beginning of each of these types, it actually tells you what the ATP is generated from. So substrate level phosphorylation just simply says that we're going to generate ATP from some type of substrate or some type of substance. So we could be breaking down some type of molecule and using that to generate ATP. The second way that we generate ATP is through oxidative phosphorylation. So again, you look at that first word, oxidative, we're going to use a series of redox reactions. So oxidation reduction. These are going to occur in a respiratory pathway where typically oxygen is involved. And then the third way we generate ATP is through photophosphorylation. So if we remember that photo represents sunlight, well then that tells us that photophosphorylation, it allows organisms to generate ATP by utilizing sunlight. So this is typically going to be reserved for photosynthetic organisms. So those that have chloroplast, they have that ability to capture sunlight and then to actually use it to make energy. So just a quick reminder of some of the things that ATP does. So we use respiration, we use fermentation, we use photosynthesis, or some other types of chemoautotrophy. From there we generate energy, and we know that ATP can do lots of different types of work within cells. So we know that it can do chemical work, so it can help us actually build or help microbes to build the things they need, like carbs, proteins, nucleic acids, lipids. It also can be used in transport work. So we know that active transport, moving substances from low to high, requires ATP. So that's a pretty important thing. And then, of course, if you'll recall, a lot of microbes have appendages, right? So they have flagella, they have fimbriae, they may have pilus or pili, and so it's going to require energy to actually use those appendages, actually to move those appendages. So ATP serves several roles within cells, which is why we have basically devoted this entire chapter into talking about how cells generate ATP.